Let's start today's session of innovative approach of structure-based drug design. Today we have 3 speakers. The first speaker is Natalie Tatum. Natalie is a senior research associate in the Cancer Research Horizon Newcastle Drug Discovery at the Newcastle University. Prior to joining Newcastle, Natalie completed her PhD at Durham University in virtual screening against antimicrobial targets. And Natalie's work is focused on structure-based drug design and development. Thank you, Natalie.
Thank you very much for that lovely introduction. So I am going to be talking about drug discovery, but not in the same way as we heard yesterday from Rob. I'm going to be talking about a technology build for target validation. So that bit before we get to actually trying to develop a drug, how do we know that we're going after the right kind of interactions? And this is really trying to get us from fragment maps to fundamental biology, which is kind of backwards to how we usually think about things. So this started in some ways from an observation when we did crystallographic fragment screening, as I'm sure many of you have done and probably have made similar observations, that fragments tend to cluster at sites of importance or kind of functional relevance. So this is a study that we published earlier this year where we were looking at CDK2 cyclin A, so a cell cycle complex. And this is data from Ian Hope, who is one of those rare people who is both a trained medicinal chemist and a trained structural biologist, something of a unicorn, I think. And so what we found when we soaked the Fraglite library in particular, so that's available at Diamond, it's a 31 fragment library that was developed in Newcastle. They cluster very specifically, richly in these sites of protein-protein interactions. So that could be the sites of phosphorylation, where the target proteins are binding. It could be sites of regulation, so phosphatases bind to regulate this complex. Or it could be on the cyclin site, a very specific localized cluster where substrates are recruited. And so we got to thinking, if you're observing these kind of things, say for a completely novel target with unknown biology, how would you go about investigating what that site was actually doing? You could use a tool compound. For a lot of these kind of things, they don't necessarily exist. It's quite an endeavor to go about developing one, as we heard yesterday. And they don't necessarily always end up or have the kind of potency and selectivity that you might be needing to really tease apart complex biology. So we often rely on cell-based methodologies. So you could knock down your protein of interest and see what happens. You could introduce mutations to affect the function, but these are quite invasive and don't really recapitulate what a small molecule binding event might be in terms of the kinetics and dynamics of that. So we got to thinking, if you're trying to investigate a protein-protein interaction, maybe you should be using a protein as your tool. Now, protein design is not a new field by any means. So we can really kind of trace the roots of de novo protein design, so creating a protein from whole cloth almost, back to the early 90s. And so there was some really early seminal work, and I've pulled together a couple of different resources here for references, looking at largely helical structures, right? Because we kind of know what a helix should be in terms of sequence. And so we see, as we go through sort of the 90s and the early noughties, the development from helical proteins, completely designed from scratch, through to larger assemblies, again, largely of helical proteins, but not exclusively. And so this was work that was pioneered by DeGrado and colleagues, and then David Baker, in using fragments of proteins to build new proteins. And this continued in a largely exponential kind of way, paralleling computational power for physics-based methods, until you get to around about 2022, which, as we all know, there was a big shift in terms of protein structure prediction. But it also led to the development of these tools where we can use this information and use the PDB, learn from it to predict completely novel proteins. And I've highlighted just a couple of papers of things that you might have heard about in this field. So we have RF diffusion, which is what most of my talk is going to be about. And this is from the work from the Institute of Protein Design. We have Bindcraft, which is a pipeline which includes RF diffusion, but is kind of more of a one-shot approach. And then the most recent is BoltzGen, which takes a slightly different approach to training compared to RF diffusion, but is really focused on trying to minimize the energy of a protein. As I say, I'm going to focus on RF diffusion, and RF diffusion is a diffusion as the name implies, based process where it was trained on the PDB, progressively noising those structures with Gaussian noise until it is essentially indistinguishable from what you might consider to look like a protein, and then trained to then denoise back towards a protein structure. And there is a long history of using animals to exemplify various concepts in crystallography. I'm going to use cats. So we have this lovely majestic ginger cat, and we're going to progressively noise him. In this analogy, he is our protein structure until essentially kind of indistinguishable, that we wouldn't necessarily say that there is a cat there. But then you denoise, and what you end up with is another cat, but it's not the same cat. It looks different. And this is essentially what the process of RF diffusion is, taking this noise and denoising towards a structure. In practice, a pipeline looks a bit like this. So here we can see that we have our static protein. So this is our target, the thing that we want to design a binder to. And it's held in place by a small motif that we're keeping static as a kind of scaffold around which to build a protein. And we are progressively denoising from this, as it comes around again to the beginning, very noisy start point here, which you would agree does not look promising, towards something that looks like a protein structure. So this is generating backbones that are entirely glycine based, apart from that bit that we've given the sequence as a starting structure. And then we graphed through protein MPNN, which is another machine learning method, sequences that are compatible with that backbone. And then in most common pipelines, then you pass these to AlphaFold, which co-folds the two things together and gives you a prediction of what that should look like. You only really get metrics out that give you quality of the fold. So we've added some things onto this kind of common pipeline where we can have a quick snapshot of how good these designs might look, whether that's looking at the quality of the fold and the interface through the PAE and the PLDTT from AlphaFold. The RMSD, so how close the AlphaFold prediction of that complex is to the original design of the backbone. We can look at that per residue. So you can see here around our little static motif, it's not particularly good, but towards the end of the protein it is. We can visualize that with the kind of standard AlphaFold coloring, and we can do principal component analyses to look at the different designs and how different they are. We can do some independent virtual validation. So often we are looking at binders to complexes. So if you're looking at multi-domain proteins or larger complexes, then you can do some independent scoring. Does your protein that you've designed stay where you designed it to be? And we can also introduce some additional scoring. So we regularly use IPSE, which is a way of qualifying how good these predictions are for binders, and also looking at the interfaces. Does this interface look particularly confident through AlphaBridge? And we did an initial proof of concept on this, looking to see whether or not we could distinguish isoforms of closely related proteins with binders. And so other cell cycle cyclins, cyclin D3, we were able to design a protein which confidently pulled down both CDK4D1 and CDK4D3. So this one's not particularly specific, does bind with a reasonable micromolar affinity. As a first shot, pretty confident. We then designed these ones for D1, and a side story which I can't go into, but it's very important what starting structure you're using to design against. We are able, in a very isoform specific way, to pull down 4D1, but not 4D3. And so that opens up the opportunity for very specific protein tools to interrogate differences in very closely related proteins. But I'm not going to be talking about cell cycle proteins today. I'm going to be talking about transcriptional proteins. So these are 3 of the transcriptional cyclins, which are involved in orchestrating the transcriptional process from initiation to a protein. You might know cyclin H better as part of the CAC complex. So this partners CDK7 to initiate transcription. And further along, cyclin T interacts with multiple proteins to elongate that transcription. Cyclin K also has an elongation role. But unlike these other 2, we didn't really have much information about what cyclin K interacts with to perform this role in transcription. What we did have, and obtained 2019-ish, was a beautiful fragment map of cyclin K. And you can see we have these 2 clusters on the C-terminal fold, which seem to be more densely populated than the rest of the protein. We very imaginatively called these site 1 and site 2, and I'm always going to show you site 1 on the left and site 2 on the right. You can see these kind of analogize how MAT1 binds to cyclin H in the cat complex, and how partners of cyclin T interact as well. So we thought that actually these might be some sites of functional relevance. The only hints we had as to what those might be was a paper that came out in 2018 and a paper that came out last year, which suggested that cyclin K interacts with CET1A, which is part of a transcriptional complex called COMPAS, and CDC73, which is part of a different transcriptional complex called PAF1C. All you really need to remember is CDC73 and CET1A. So could those be our sites of interaction? Well, we did some AlphaFold modeling and tried to fold the region of CDC73 and CET1A that were implicated with cyclin K. And you can see for CDC73, we have quite a bit of high confidence around that site 1 site, but it doesn't really overlap site 2 or where our fragments bind. Whereas CET1A is quite confident in site 2, but quite poorly confident, although it does seem to at least spatially put that bit of sequence around site 1. So this is a little bit of a kind of indication that these might be our sites of interest for these particular interactions. And when we compare how the fragments bind to these particular models, we can see that both models, though one is considerably less confident than the other, both put a phenylalanine, which overlaps very nicely with one of our fragment hits. The CDC73, which is much more confident, also kind of analogizes a different fragment hit. And over in site 2, the only bit of CET1A confidence overlaps quite nicely with this entire smear of fragments, but particularly exemplified by fraglite 1, where we see these hydrophobic burial in this dimple down here, occupied by 2 fragments, which is where the side chains kind of bury in there, and also up here where we have some more polar interactions. But this was well before we got into the design bit, which I promise I will come back to. So we had to use these cell-based methodologies to investigate this. So we went from our fragment map and we designed some mutants at these sites. So we have a site 1 mutant, which is largely just about changing some of the charges to prevent some interactions there. Site 1's quite hydrophobic, so there's not a lot that we could do without risking too much perturbation of the structure. So we were quite conservative there, whereas site 2 was quite charged, so we were able to make more drastic changes. And as a control, we also made a mutant that would prevent from CDK12 from binding, which could be used to look at whether or not any CET1A or CDC73 interaction was specific to the cyclin K. So a very talented PhD student, Arwa Al-Shambari, was working on this. She's frantically writing up and finalizing her thesis now. So she took these mutants and the wild type and transfected them into HEP293 T cells, which are kind of a basic standard set of cells to work with. And these have a 3 flag tag on them. So we can pull down on our wild type or our mutant proteins and then Western blot for things that we're interested in. So here, I'm just going to make sure that that's, yeah. So here we have our wild type cyclin K, which you can see pulls down CDK12 as we expect, pulls down CET1A, which is exciting. And we have a nice blot for the presence of the flag Ka. Our site 1 and site 2 pull down CDK12, much less on the CET1A, very exciting. And our knockout site at CDK12 still pulls down the CET1A, so that's very specifically a cyclin K interaction, but we don't see any interaction with CDK12, so that's a nice little control for us. We took these forward for mass spec, because if we're looking for very specific proteins when we do a Western blot, we want a more global look, right? So this is what the site 1 pull down looks like. And we're looking for things that are decreased with the mutant. So things that are reliant on that mutant site to bind to cyclin K. And so you can see here on the right, left in blue, we have detected components of both these complexes that the modeling suggested both bind at site 1. So everything we detect from PAF1C and COMPAS, with the exception of this DPY30, which is a very short protein, we see 1 peptide from it, so statistically it kind of got weeded out. So it would suggest that site 1, identified by our fragment map, is a shared regulatory site for these two complexes. But it would be kind of remiss of me if I didn't address the elephant in the room, which is this large plume of red stuff. And so this actually seems to contain a lot of stress-related proteins, heat shock proteins, that kind of thing. So either interfering with this site, which is a clearly a shared regulatory site for a lot of different transcriptional functions, has a drastic effect on the health of the cell. Or it could be the perturbations that we made at that site made for a less stable protein, and the whole thing's just kind of pulling down a lot of chaperones to keep it as happy as possible. In contrast, site 2 is exquisitely specific for that set1 compass complex. We don't see any other complexes in that, and the gene ontology analysis backs that up. We only pull out this methyltransferase complex. So it would suggest that site 2 could be a very specific way to interfere with cyclin K function. How do we know that that has any effect on the cancer biology that we're interested in? We don't. So this is where you would come in with trying to really understand which settings are important, which types of cancer might be important or impacted. And as I say, CDK12 cyclin K has roles across various things, not just phosphorylating targets, but also being involved, for example, in the DNA damage response. Now, this is kind of a prime place that we could interfere, but we need a tool to really start investigating that biology. We don't have a tool compound, certainly not for a site that has not been studied before. And we have initiated some of those cell-based methodologies. So we're looking at rescue experiments where you kind of knock down the endogenous cyclin K, introduce your mutants, and hope that you see some kind of response there. But which cell lines do you choose? Which settings are you interested in? That's a lot of technical parts to maneuver. It's a lot of time, it's a lot of cost. A protein probe could give you a more authentic background because you don't need to modify the cells in any way. All you have to do is transfect in this binder. You could assess a wide panel of cells, so you wouldn't necessarily be restricted to one particular cancer type. You could have a look at different cancer types, different pairs of cell lines. And that could also be very versatile for mechanistic studies with different tags on that binder protein. So we went ahead and did one. So what we did with this was use both the fragment map and our AlphaFold models. And this was one of 3 master students that we had with us last semester, who had, we thought, kind of the riskier project, right? There's no structural information really. We have a AlphaFold model with some kind of high-ish confidence for that small bit of the set 1A model. But you know, it's a master's project, it would tell us something. And so we went ahead and used that little bit of set 1A helix that we were reasonably confident about, which overlaid with our fragment map to scaffold a protein. We knew which hotspot we were interested in from that site 2, and we could also block site 1 with a small motif from that CDC73 to make sure that our designs stayed encompassed around that site 2 site. And we could do some virtual validation. So we ended up picking 4 sequences, which is not a lot, but we had pretty high confidence that these would be potential binders. They have quite low PAEs. They are quite confident in the fold prediction. When you do it independently, it doesn't go anywhere near either site 1 or CDK12. It's very constrained to that site 2. It's very high confidence. We were pretty optimistic. So one of the last experiments we did before Christmas was to recombinantly express these and then do some SPR. Every single one of them binds. So we had these 4 designs, and they have slightly different kinetic behaviors. Largely speaking, these 3, they go on, they don't really come off. This last one, at least, is quite faster on and a slightly faster off rate, which is quite useful. And when you try and do some of the fitting to get affinity measurements, these are all coming out around about low nanomolar or better. Eagle-eyed SPR aficionados among you might have noticed there's something different about this particular one. So the lowest concentration, it goes on, it doesn't come off, and this x-axis is slightly different as well. This goes out for 10 minutes. It doesn't come off. And then as you increase the concentration, actually you've just got fewer sites to occupy. So the titration goes the other way around. And our instrument was not able to actually assign an affinity for that. So what we've done is we've ordered this one, MW1, and this one, MW4, for mammalian cell expression to basically put into this system and have a look and see what that site 2 is actually doing when we interfere with it in various cancer types. And so we're really excited about this as a potential tool for target enablement. For structural biology target enablement, which is a large part that tends to be kind of underrepresented sometimes, it can be quite difficult to get the reagents or the structural information that you need. So we can use them to increase mass for cryo-EM, increase asymmetry. So for example, with the CDK cyclin complex, that can sometimes be quite difficult, both in terms of size and resolution to determine where the partners are, or using them as bait on scaffolds to specifically pull your protein, either maybe out of cell lysates, for example, or in more specific recombinant systems. You could use them to stabilize particular conformations. So we know we can get isoform specificity. Can we select for active versus inactive conformations to open up different conformations for fragment screening? And also the kind of implication for cellular probes is really exciting. So at the moment, we're going to set them up with a self-cleavable GFP. So we're able to see that it has been transfected and expressed, but it's not actually going to interfere with the binding. And then we can look at a very clean biological system. Or you could keep it on. So we'll be working with people to use them as probes for immunofluorescence, for example, to look at very specific interfaces. You could use them almost the way you use antibodies for very specific pull downs, for example. And we're quite excited about turning these into bioprotecs. Now, a lot of these are necessarily new concepts. It's things that people are doing with nanobodies, for example, but you can make very stable, very highly expressing proteins in this manner, which will accelerate projects through this target validation stage to really weed out the projects that don't seem to have the biology that stacks up and prioritize the ones that do. So with that, I just want to thank everyone who's been involved in this work. As we've heard, it's very much a team science. So everyone who's been involved in the structural biology at any point. The cell biology was very much driven by Arwa, and more recently Vicky, who's taken this into specific cancer settings with these rescue experiments. The mass spectrometry was done in-house at Newcastle with Matthias Trost group. And we had 3 students, each working on a different transcriptional cyclin that I showed you. And it really showed that when you've got some really excited, engaged students, they can do some really phenomenal, groundbreaking work. And of course, it would be remiss of me not to thank the beamlines who've contributed to the structural work. One thing I do have to remind myself to mention, we do have a couple of technical positions coming up in Newcastle that currently being advertised internally, but if they go unfilled, then they will be advertised externally. So if any of this has piqued your interest in working at Newcastle, keep an eye out for those. And I'm happy to take any questions.
Thank you, Natalie. There are any questions? Can see you.
Oh, there's one over there.
Oh, sorry.
Yeah, so quick question. In the beginning, you show that you're designing proteins that bind to very, you want very specific binders to things versus some of them. Do you do anything in the computation to actually account for that? Or do you just make binders and then hope that they pull down just the target that you want?
Not specifically, but what you can do certainly at kind of this stage, once you have some things that look quite confident for the thing you're interested in, what we encouraged the students was to basically swap sequences and try them against the related cyclins, because they are very similar in structure. There is some conservation around them. If I go back to here, not even that one, if I go back to here, so that they could have an idea of what that at least virtual validation might look like. And then when we got to the experimental part, we were screening all of the binders against all of these different cyclins. So our cyclin K binders are very specific for cyclin K. They don't bind either of the other two.
I see. I guess what does the computation give good? I mean, is that worth running that experiment computationally? Like, does it give good feedback on then what works experimentally, or are these methods not so good at picking out like the AlphaFold prediction? Does it actually predict these subtle differences?
There are some correlations between the IPTM and roughly an affinity. The IPSA score has a cutoff below which it's very likely not to be a binder, very likely to be a binder. At the moment, there's just not enough data on the binders and their affinities, and we've already talked about different assays for different affinities to really drill down into what might might inform that, but it's something that we're really interested in, because especially if we are trying to make these as selective as possible, doing that as early and building that into the workflow is very important to us. Okay.
Thank you for a great talk. I was wondering, the first part where you use the fragments to guide sites of interest, have you used other ways of identifying sites of interest, whether that be conservation or hotspot analysis? So is it possible to skip the first step, or make that first step, do that differently, that might tell you that there are sites of interest without doing the fragment screen?
Yes, I think it would be potentially a bit riskier, maybe, but it is possible. I mean, these are sites that are conserved. Among the cyclin K, for example, site 2 is conserved within cyclin Ks, doesn't really seem to be conserved across the family. So it certainly is possible, and you don't even, what we've noticed, you don't necessarily need to have a scaffold to start off with. You can just do this kind of thing blind. What we're interested in is kind of going from that experimental confidence, a kind of fuzzy computational step where we can kind of build a bit of theoretical confidence and then come back to the experiments. But there are other ways in terms of hotspot analysis and conservation, sequence conservation, that could help you guide if you weren't able to necessarily get recombinant protein to start off with.
Hello, very interesting approach. Have you tried any of the binders from your initial screen to interfere with the interaction? For example, the set1 interaction?
That is literally what we're hoping to do in the next 2 or 3 weeks. So yeah, so what we're going to do is, if I show you the Western blots, we're essentially going to kind of repeat this, but rather than introduce our mutants, we're going to introduce the binder and see whether or not in the presence of the binder, we reduce that set1A interaction, which would be, kind of the first experiment to check that we're actually hitting on target where we think we are, but then also repeat the mass spec. So in the presence of the binder, do you also see interference with the set1A complex? And do you see anything else? And additionally, if you pull down on the binder, do you only see cyclin K? And those are the 3 experiments we desperately want to get done in the first half of this year to make sure that what we have is a very robust tool.
Cool.
Okay, thank you so much, Natalie.